Hi guys, welcome to my MCAT channel. Today let's talk about hydrophobic interaction chromatography. So chromatography is a pretty high yield topic on MCAT. So most students are clear about like size chromatography, ion exchange, affinity, or thin layer or gas chromatography, right? But this topic, hydrophobic interaction chromatography, is really a type of chromatography that is not super emphasized on the MCAT textbooks. And I've never seen a video where it explains it super well from the MCAT perspective. So today, I want to make this video for you if you're just obsessed with MCAT content as I do. And then if you really just want to get one question correct where 95% of the students miss, then let's dive into this, all right? So, okay, like I said, hydrophobic interaction chromatography is a type of a liquid column chromatography, okay? So we see our column here, and then our liquid, our solvent is really most of the time water. So let me draw a water molecule for you, okay? H2O, all right. And then what about the solute, right? The solute is the protein, okay? The proteins are floating in this column. And then the purpose of this lab technique is really to dilute the protein of our interest, right? We want this beaker to collect the protein of our interest, given the different properties of protein, because some proteins are hydrophobic, some proteins are hydrophilic. We're gonna see which type of protein is gonna elute first, okay, into our beaker. All right, so let's come back to our water, okay? So we have our solvent water here as our liquid, and then the water is really always interacting with the solute protein through hydrogen bonding, right? So let's say this partial positive hydrogen is attracted to the partial negative part of the protein, okay? All right, and then this partial negative oxygen is attracted to some partial positive part of the protein, right? Okay, all right, so it's really stuck there, right? I mean, how do we elute the protein out if they're interacting with the water, right? So scientists, they figure out that they come up with those beads, right? Those beads are the stationary phase, meaning that they're not mobile, right? They're not moving. So they have a stationary phase where they coat these beads with some hydrophobic coating, okay? So as you have imagined, this hydrophobic coating would want to interact with the other hydrophobic stuff, right? Okay, so our purpose is that we want those are hydrophobic protein, okay? Let's say there's a hydrophobic protein, okay? Protein A. We want this hydrophobic protein to bind to this hydrophobic coating of the beads, right? So this is a hydrophobic binding. So that when most of the hydrophobic proteins are binding to this stationary phase, the mobile phase can have the hydrophilic proteins elute out first, right? Okay, so that we can collect, oh, these are the portion that's hydrophilic. These are the hydrophilic proteins that's what we're interested in, okay? All right, so this is the idea, but still, like I said, you can't ignore this hydrogen bonding interaction, right? I mean, the proteins are stuck. They're not like, oh, I can go just the hydrophobic one, let's just go bind to the stationary phase. There's this hydrophobic coating, let's go, right? They can, they're attracted to water, right? So what's the way out, right? So the way out is really salt, okay? So as you can imagine, when we dissolve salt in the solution, like water solution, right? We would, let's say NaCl. That NaCl will really break down into a cation and an anion, a positive thing and a negative thing, right? So that when we have this positive or negative thing in my solution, when they dissociate, right? When the salt dissociates, then my water will be attracted to salt because now this cation is a fully positive charged thing, right? So that my partial negative oxygen would want to bind to this cation, right? My partial positive hydrogen is going to ditch this partial negative protein part and then really bind to the negative anion, okay? All right, so this salt interacting, I mean, the dissociated salt particles, right? The cation and anion, they interact with a water molecule. It's really called what? Hydration, right? Hydration, remember, it's a special type of solvation, right? Hydration is in terms of a solvent being water, it's a hydration, right? Meaning that, okay, the solvent water, their partial positive and negative atoms are attracted to these fully charged molecules, okay? All right, so this is how salt can attract, or we can say distract, water, right? So that water binds to those anion cations, okay? So water binds to these cation and anions so that the hydrophobic proteins are free. Now they're free to bind to the hydrophobic stationary phase, okay? So that the hydrophilic ones can be eluted out first, as you see here, okay? I put a hydrophilic protein elute out, okay? All right, so this is how this works, right? It's pretty smart. Okay, so on MCAT, what if they ask you, they say, okay, what if now I'm gonna reduce, okay, when I reduce the salt concentration. Okay, so this is really intuitive actually, right? Because when you decrease the salt concentration, you're basically compromising, weakening this system, right? You are not like letting all the proteins free, right? You're not letting all the proteins free so that it really compromises the hydrophobic proteins binding with the hydrophobic stationary phase, right? So in this case, our elution will be more of like a mixture of hydrophilic and hydrophobic proteins. It won't be such like pure hydrophilic protein if you were to give it enough salt concentration, right? So the effect of decreasing the salt concentration would really increase the hydrophobic elution. Okay, all right, more hydrophobic proteins will be mixed in the elution, will be eluted, okay? All right, so that's it, okay? It's a fairly, I mean, it's not super straightforward, but it's durable, okay? All right, and then one last thing, I have two more things to say. Okay, so one more thing is the salt that you encounter on MCAT regarding this hydrophobic interaction chromatography is usually not NaCl, okay? It's ammonium sulfate. Okay, it doesn't matter like how fancy the name sounds like. They disassociate into one positive thing and one negative thing, right? Further interact with water so that the proteins are freed to bind to the hydrophobic ones can bind to the hydrophobic stationary phase. So the mobile phase as the hydrophilic ones can elute out first. Okay, all right. So that's the logic. Okay, so let's dive a little bit more to make it a little bit complicated, okay? I mean, still the same idea, but just different ways they can phrase it. They can test you on this, okay? So let's say now I have this anchored, right? I have this stationary bead that's coated with my hydrophobic coating. So I'm binding to, let's say, protein A, right? This is not hydrogen bonding. This is really a hydrophobic interaction, remember? So this is like a London force, right? Something like that. Okay, and then in another setup, in another column, I also have these hydrophobic coating, and then I'm interacting with protein B, right? So as you can see, like I drew more interaction. I drew more lines here indicating that protein B is binding more strongly with this stationary phase than protein A does, right? So if the question asks you to choose, okay, so according to the graph, which one, A, B, I mean, let's do this protein 1, okay? I don't want to confuse it. Protein 1, and this is protein 2. Okay, so protein 2 is A or B, okay? Pick one. So let's say A is like A, E, D, okay? And then my answer choice B is like A, D, okay? So which one is protein 2, A or B, right? Which one would you want to pick, right? You want to pick B, right? Answer B is your answer. Because why? As you can see, protein 2 is more strongly binding to this hydrophobic coating. So hydrophobic thing interact with hydrophobic thing, right? So then you according to the amino acid sequence, you see, oh, the more alanines in answer choice B, right? Here I have negative thing here. So A is really more hydrophilic than B, okay? So that protein 2 that is more hydrophobic is, of course, answer choice B, okay? All right, so that's another way they can test you, right? And then also we can, another way is, I'm thinking that we say, okay, so protein B is more hydrophobic, at least on the surface level, right? Than protein A, it binds more tightly. So it would need more help or less help from the salt, right? It would actually, this protein 2 is actually needing less help from the salt. You can kind of decrease the salt concentration a little bit, right? Because its interaction is just so strong, you know, it's easier for protein 2, this more nonpolar one, more hydrophobic one, to bind to my stationary phase. So it really needs, so if I were to draw the salts in this column, then I would just like draw like a 2 NaCl. I mean, it's ammonium sulfate that I told you, but just to easier this process, right? Whereas for protein 1, I would need a ton of NaCl. Okay, all right. So this always, I can imagine that they can test you on this. If you have any more questions, please leave me a comment, and I hope it's helpful.